So I'm going to talk about epilepsy therapeutics with a focus on gene therapy and a focus within that, where possible, on KCNQ2, because right now there are not a lot of gene therapies for the genetic epilepsies, but they are emerging and it's an exciting time. So I just want to start out, I do have conflicts, although I will say none of the companies I'm involved with have anything near something on the horizon, I would say in the next 2 to 5 years, although I am hopefully consulting for some companies like Ultragenyx that may have therapies for CDKL5 deficiency disorder within 2 or 3 years. That would be my hope, but we'll see. So to begin with, and this is something I think the families at the summit have been through, but it's important with genetic epilepsies because many parents may have had a 10-year-old child who went through a gene panel 5 years ago, others who may have a 40-year-old or 30-year-old child who may now be living in a group home. And they kind of feel like they did an extensive diagnostic workup a long time ago. But I would really encourage, probably not so much the parents at the summit, because I think you know what your child has, but friends of yours who may be roommates in group homes or other children with neurodevelopmental delays, that you need to push for genetic answers, because that's the only way we can find targeted small molecules and gene therapies as they now come to occur. So it really opens a lot of doors having answers. There are, of course, a number of clear implications therapeutically where, for example, I'm not going to go through the list, and these slides you're welcome to share with the audience later, but for GLUT1 deficiency, which is due to a specific SCLC2A1 gene, ketogenic diet is magical and works incredibly well. Many other disorders, including KCNQ2, not every mutation, but a lot of them do very well with sodium channel blockers. And the list goes on. mTOR inhibitors or disease-modifying therapies for the tuberous sclerosis complex genes, and possibly for some of the other related genes. And for genes like SCN1A that causes Dravet syndrome, there are at least 3 medications that are actually specifically approved for that syndrome. So we're getting closer, and as I said on the bottom here again, KCNQ2 epileptic encephalopathy, based on the experience, overall, probably the best category of medications are those that block sodium channels like carbamazepine, oxcarbazepine, and related compounds. So this is a slide from a summary by Kirstein's group and others of a large number of cases of KCNQ2 encephalopathy versus the benign early life form of the disease. And there are some, I'll just focus on a few of the statistically significant differences. The frequency of de novo mutations is far higher in the early onset epileptic encephalopathy, 83% versus 26% or so in the benign cases. So if it's something neither parent harbors, that it's a new mutation, it's more likely going to be a more severe type of epilepsy. And the same thing is true for intellectual disability. Those that are new mutations, not present in either mom or dad, have extraordinarily high rates of intellectual disability, whereas those that are inherited, typically if parents are going to have the gene, one of the 2 parents, and is going to have children, they're usually intellectually normal or quite close to it. Other things like sex, there is no difference for. So, aggregating large numbers of cases like this is very helpful. We can look at the types of mutations that occur and where they are. And again, very informative. And I think your community has been excellent in aggregating these cases. And as we look to the future with gene therapy, I would say KCNQ2 is going to be similar to some of the other ones. You as a community have a tremendous amount to add, specifically around the natural history. You know your children. Doctors see them for 20 minutes, an hour at an office visit, but you live with them. You know the nuances. It may turn out what many companies and investigators have used as the primary outcome measure of seizures, hopefully may be fairly well controlled in a lot of your kids. So you might have an ASO or a gene therapy that we'll get to that might truly be miraculous. And it might be after 6 months or a year, or 18 months of therapy, it substantially improves cognitive function, behavioral function, motor function. But if you can't measure it, you could have the golden answer in your hand and it could slip through your fingers. So we need to have measures that are sensitive and reliable. And we all have to be honest with each other. These things are hard to do, and we need to be objective. So again, it's something that we need to work on as a community, but the time is now because gene therapies will be out there soon. So this slide just goes to show that not so much for neurologic disorders, although Speranza over here is a treatment for spinal muscular atrophy. It's an ASO therapy, and that is on the market. But there are now a lot of approved gene therapies that are out there. There are others that are being emerged for cancer and other disorders. But this is a field that will be, it already is expanding rapidly. The slide's from a year and a half ago, but the list just keeps growing and growing rapidly. We, unfortunately, for brain disorders, have more challenges. If it's a blood disorder like hemophilia, it's sadly one of the easier ones because we have such great access to the blood. The brain is protected by the blood-brain barrier. You eat poison mushroom, you may vomit, you may be sick, but you won't die because the compounds that are toxic from that mushroom typically won't cross this barrier. So the barrier is there to protect us, but it also protects us against potentially beneficial medications or gene therapies. So some of the other challenges are that we need to package a gene therapy, especially if you want to replace a gene. Someone has a defective copy of KCNQ2 and you want to introduce another one. It's a huge gene. And right now, the viral capsids, literally protein envelopes that we package these in, can only carry such a big load, as I'll get to. If you use viral vectors, our immune system responds to them, and you may only have one shot on goal. The second introduction of that vector may provoke a terrible immune response. The dosing, it's really hard to get enough of a gene therapy into the brain. ASOs are special. They have long half-lives, and you can introduce them through the spinal fluid in the lower back. But other gene therapies, it's really a challenge getting it through the blood-brain barrier. And then we've got 80 or so, 90 billion brain cells. How many need to be changed by the gene therapy? Is it 5%? Is it 20%? We have big brains, and monkeys are good models for many things, but they're not good models for the size of our brain. Another thing we worry about with any gene therapy is off-target effects. Obviously, we want to improve the function of a diseased or mutated gene, but we don't want to cause changes in healthy genes or introduce too much of a dose that could be detrimental. And then how long does the effectiveness of a gene therapy last? ASOs are being dosed, for example, for spinal muscular atrophy every 6 months or so. We'll have to figure out what the right dosing is for every disorder. And then sometimes you may upregulate one thing, but the gene network is complicated. Biology has evolved homeostatic mechanisms to keep everything balanced. You work out, you do vigorous exercise, your body temperature will go up, but your body has many measures to lower your body temperature when you do exercise, like sweating. So keep in mind, we can affect things, but biology is usually 1 or 2 steps ahead of us. So I just want to discuss the kind of genetic mutations that occur. And again, it's obviously we're all here at the summit, and it's for KCNQ2, a very specific gene, but you may have similarities with your child's mutation with families that have SCN2A or 8A or 1A or many other gene mutations. So the wild type is essentially someone has 2 copies of the normal gene and they make 100% of the normal protein level. There are autosomal disorders where there may be 2 abnormal copies, one from mom and one from dad, in which case those can be very severe. There's haploinsufficiency, which affects over 500 genes and causes disorders. And that's where there's 1 good copy and 1 bad copy. So the individual is running on 50% of the normal protein function. There are a category of gain-of-function mutations where, for example, a sodium channel or potassium channel may open for a certain amount of time. If you have a mutation over here, a haploinsufficiency, that channel may not open at all, in which case you have a loss of function or haploinsufficiency. One of the proteins is essentially not working well. In a gain of function, it's the opposite. That channel may open too long. And so instead of not getting enough sodium rushing into the nerve cell, you may get too much. And so the drugs that may be very effective, for example, in treating a haploinsufficiency, will make a gain of function worse. And then there can be a dominant negative where there's a mutation in one copy of the gene, but it works to impair the function of the target. And so it actually overtakes the normal copy of the gene. And instead of having 50%, you could be running at 0 to 30%. So these are just broad categories. We'll get to more categories. But keep in mind, just because everyone in the room has a KCNQ2 mutation, some of you may have, for example, a premature termination or stop codon, and that represents, I think, about 12% of the mutations in KCNQ2. And those mutations may be treated the same, whether they're in SCN1A2A8A, whether they're in CDKL5, whether they're in a large number of different genes. So you actually might share more in common and potential gene therapies with a parent of a child with 10 other different gene disorders if they all share the same type of mutation. So keep in mind, your relationship is not going to be tied to just one gene. It's also going to be tied to the type of mutation your child has. So here are some of the potential therapies that we can think about in broad categories, and I'll just run through them briefly and we'll spend some time on each. There are, for example, small molecules to read through a premature stop codon. So again, as I'll get to, every 3 letters in the DNA codes for an amino acid, and 3 combinations of 3 letters code for stop codons instead of an amino acid. So if you're in the middle of a protein messenger RNA chain that's going to read through 1,000 amino acids to make the protein, and the amino acid 500 or 50 is supposed to be alanine, but a single letter changes, and it changes it from alanine to stop here, you will get haploinsufficiency. That one copy will be terminated, the cell will recognize it's defective, and essentially will chew it up way before it gets to the ribosome to make a protein. Antisense oligonucleotides, or ASOs, are a leading and current form of therapy for many disorders. And there is a company that has created a charitable foundation called NLORM, and they will actually take individual cases if they're rare. They don't want to go for something that's going to treat everybody with KCNQ2, but if there was a specific mutation that was rare, and they felt that other companies were not going to go for it, they've taken that on as a pro bono thing, because they were the company that developed some of the successful ASOs and other disorders, and they're trying to give back to the community. Transfer RNAs are bits of RNA that specifically will transfer 3 letters in sequence that are attached to an amino acid. So it's the way that our ribosomes, when they have a strand of messenger RNA that says ATA, they'll recognize the complementary base pairs, and attached to the complementary base pairs is the amino acid. And that's how the ribosome literally translates the messenger RNA strand into a protein with amino acids. But these transfer RNAs could be used to read through premature stop codons, because you could literally bind to a premature stop codon with a transfer RNA and substitute an amino acid, and thereby, instead of cutting a protein that should be 1,000 amino acids short at 50 or 500, you'll have a 1,000 amino acid protein, and one single amino acid will be changed, which often does not change the protein's function. There are also transfer RNAs that can be used to increase or prolong messenger RNA production, which will result in more protein being made. So there are some nice opportunities with these to increase production of genes that are haploinsufficient, or to read through premature stop codons. Regulatory elements, or little bits of DNA, my metaphor for them is that if lights in your ceiling are like genes that make proteins, the proteins are like lights. We all know to get lights on in our rooms, we typically have switches on the wall that turn them on and off, and dimmers that turn them up and down. The regulatory elements are like they are switches. They are little bits of DNA that typically occur before the gene, sometimes between the first two exons or pieces of a gene, and they regulate. Without those regulatory elements and promoters, the gene could be there, but it will never get transcribed into a messenger RNA. CRISPR is a way to edit genes and can fix a large number of changes in a gene that can increase functional protein expression. And CRISPR gene editing, there are many large companies doing this. Unfortunately for us, getting these CRISPR gene editing enzymes into the brain safely is a huge challenge. So almost all the companies are going after the heart, the blood, the eye, the liver, places where they can deliver it pretty easily. I don't believe any right now are going after the brain, although our group has started to work on it with the hope that the delivery system and safety will get there in short order. And then finally, there are companies and investigators doing entire gene replacement therapy. You just package a small gene into a viral capsid and deliver it and put the appropriate molecular machinery in so it incorporates into the chromosome and can be expressed to make the protein. So let's start with premature stop codons. As I said, I think this is about 12% of the gene variants causing inherited human diseases and 20% of diseases associated with single base pair changes that alter the coding regions of a gene. So they're pretty common. They're not going to be the majority of KCNQ2, but they're a significant minority, and they're a significant minority of all neurogenetic diseases. So just some basic math for a second. There are 4 letters in the DNA code. C binds with G and T binds with A, so those are the 4 letters, and they can occur in any combination of 3 letters. A 3-letter combination codes for an amino acid. There are 64 combinations of the 3 letters. It's 4 times 4 times 4 is 64. And Watson and Crick figured this out actually at a pub called the Eagle outside of Cambridge. And they were sitting one day and they were thinking, well, there's 20 amino acids and there's 64 combinations. Well, that doesn't make sense. Nature's not an idiot. She wouldn't make all these extra things. And it turns out there's a code within a code, and the code makes a lot of sense. The amino acids that evolved early in life tend to come from the same precursors like pyruvate. So there are many 3-letter codes for those. Those that came later in evolution have far fewer 3-letter codes. And then the first letter codes for whether it was made from pyruvate or another simple base, and the second letter tells whether it likes to be in water or likes to be in fat. And so if there is a change in the 3-letter code, it tends to be conserved. The amino acid may change. First of all, it may not change at all, but if it changes, it will change into something somewhat similar. So it's a remarkable code. People thought it was chance and accidental, but nature is always smarter than we are. And then, as I said, 3 of those 3-letter combinations code for a stop codon. And there are different drugs that were discovered. The first ones were the aminoglycoside antibiotics, specifically gentamicin. It binds a specific area in the loop of the decoding center of the bacterial ribosome, which is why it messes up bacteria and why it's a good antibiotic. But it also reads through premature stop codons. Gentamicin is unfortunately a relatively toxic amino acid. You got to be very careful. I remember in my internship, you had to measure levels. If you give too much, people can go deaf. So it's not something we tend to use in medicine, except under dire conditions. But there are other compounds that have been found. So there are other aminoglycoside antibiotics that are safer derivatives. There's a compound I used, Adalurin, which is made by PTC Pharma. This was a study in premature stop codons causing either Dravet syndrome from SCN1A mutations or CDKL5 deficiency disorder. Unfortunately, it did not work in that study, but we used only 1 dose. And I was skeptical that the company knew what the right dose was before we started. And I'm not sure if that was the problem. We'll never know, but I do hope there's more research on small molecules because they are clearly a relatively easy and safe way to increase protein expression and could be a wonderful disease-modifying therapy. There are some other medications or drugs identified by high-throughput screens like clidocene, which is derived from a mushroom. It's an adenosine nucleotide analase, and again, reads through premature stop codons. And as I said, suppressor transfer RNAs, these are altered transfer RNAs with anticodons that complement the stop codon. So they recognize a stop codon, but instead of saying stop the protein, they insert an amino acid, which largely preserves the length of the protein and also the function. So these are all different therapies that are now being explored by different companies. What about ASOs? And I think there's probably a lot of interest in the community with ASOs because these are at hand. Investigators, laboratory people at Harvard, at UCSF, at UC San Diego. There are many investigators making these right now. Different synthetic devices can make them pretty quickly. They're single-stranded synthetic nucleic acid chains. They have fairly wide distribution. If put in the spinal fluid, for example, for us, they have long half-lives, so they can be dosed every 6 months in some disorders. And they depend on the correct dose of the messenger RNA transcript in the disease-causing gene. So the goal is to restore the amount of the missing messenger RNA. And they can alter the expression through multiple different ways. One of the biggest is an enzyme, ribonuclease, H-mediated or RNA-induced silencing compound, RISC for short, that increases the pre-messenger RNA decay, which decreases messenger RNA expression. And this would be the kind of thing you would use to treat a gene disorder where there's an abnormal protein that's being made. So if you had Alzheimer's, for example, and you're making too much of a protein that's getting clogged up within lysosomes and destroying nerve cells, this kind of ASO would be a way to downregulate or reduce the amount of messenger RNA expression. There are some other ones. I'm not going to go into too much detail, but there's also steric blockade, where you block the ribosome from translating. And then you can also, there are some splice site variants, so a mutation not in the gene itself, but where exon 1 splices genes. Keep in mind, they're not just little beads on a string. They are beads on a string, but the beads on a string can get broken down into 10 or 20 parts. Those parts are exons. And so a bacterial gene is continuous. It's typically a circle, and that one circle is one gene, and it's nice and simple. Our genes and our chromosomes, as you recall, straight. And those genes may literally be cut up into 20, 15, 25 pieces called exons, and between them are non-coding DNA. And so adjacent to each exon are splice sites, because it's like taking tape and splicing together, and there's molecular biology, evolution has put together splice sites. And some of the abnormalities can be in these splice sites. In addition, if there's one exon, for example, that a lot of the mutations occur with a single disease like Duchenne's muscular dystrophy, people have figured out a way to use ASOs to skip an exon. So if exon 17 is the diseased one in a patient, but really the protein will work just fine if you combine exon 16 to 18 and then do the rest of the protein chain, you can exon skip. Or if there's an abnormality at the splice site, you might be able to work around it. So there are many, many different ways to do this. And then the final one I'll mention is a way to increase messenger RNA and protein, and that is binding to the pre-messenger RNA. And I'll get to that with Dravet syndrome. That is actually the strategy being used by Stoke Therapeutics in a trial that's ongoing now. So this is to introduce you to a company that figured this out, has gone through a lot of preparation, and is now treating patients. I think we've enrolled 2 or 3 at NYU to date in what we hope is a very promising treatment. But what this slide shows, and this is TANGO, what they called targeted augmentation of nuclear core output. And essentially, if you look at the top here, you have the wild type over here and the mutant. So the mutant's never going to give rise to good protein. But what happens in the normal pre-messenger RNA for the normal gene, some of it is degraded. I'm not sure why 100% biology does it, but it makes some here that you can see the pre-messenger RNA that just gets degraded in the nucleus before it makes it out to the ribosome. And the reason it gets degraded is it has a little, what's called a poison sequence over here. And part of that is probably including a premature stop codon. What the Tango does, what their ASO does, and you can see down here, it binds to the mutant one, and that's okay because it's doesn't make functional protein, but it binds to the pre-RNA that was doomed to be degraded, and you get more of it. Instead of one strand coming out, you get two of them coming out. And that's because it locks onto the poison sequence, and that pre-messenger RNA is not degraded and is able to make protein. Again, not want to get into too much detail to lose you, but it's a fascinating, brilliant little piece of molecular biology, but it is a way, and it clearly works in Petri dishes, it works in mice, and now the big question is, will it work in our children? And we'll find out, but that trial is now underway, but it is a way to restore near normal protein levels in children with neurogenetic disorders that result from one bad copy of a gene. And this clearly, you've got to have the right pre-messenger RNA with this kill sequence or poison sequence. That's the key. If you have that in your gene too, this would be a viable therapy. If you don't, this specific ASO strategy wouldn't work. And these are just some slides, again, not to go into it, but in a single intracerebral ventricular injection of this ASO in Dravet mice, it very greatly reduced the incidence of SUDEP. The red line is survival. This is terrible survival. Up here, the blue line is dramatically improved survival. And then this is latency to first seizures. This is without it. This is with the ASO. And again, this is SUDEP incidence here as well. So you can see in the mice, these ASOs are very, very effective in survival and also reducing seizure frequency. So what are some of the other ASO strategies? And again, there's a bit of a summary of what I went over previously, but there can be targeted ones that target the RNA transcript that produce disease-causing proteins. So this is a way to shut down bad proteins. There's splice sites, as I said, that can allow you to produce less. These are all pathogenic proteins, which is a big problem in a lot of the neurodegenerative disorders. And then targeting translation start sites. So if you can make it not start, you obviously won't produce the protein. And those are relevant for some of the epilepsy genes here, SCN8A and 1A. So let's now turn to a different, and for me, a very fascinating molecular biology tool. And these are regulatory elements, the switches and dimmers that I referred to before. There are about 516 recessive disease-causing and 660 haploinsufficiency disease-causing genes. All of these would be potentially candidates for regulatory element therapy. And you can see about a quarter of the recessive genes and about a fifth or so of the haploinsufficiency genes are just too big to be packaged into the standard adeno-associated virus, which is the viral capsid that's used for gene therapy, especially in the central nervous system. So you can't have it more than 3,000 base pairs because these AVs also have to carry 1,700 base pairs to drive it. Remember I said you need promoters, you need other things. It's a little bit like engines are great for cars, but you need some other parts to make the car run. You need a gas tank, you need, I'm not a mechanic, but there's other parts as well. So this is the equivalent of those other parts. The engine's great, but you need other things to make the engine work. So basically, you're looking at about a 4,700 base pair packaging capacity for these AAVs. KCNQ2, guys, has 72,000 base pairs. It's a lot of these ion channel genes are monsters. They're big, complicated genes that go in and out of the cell membrane. So it's not likely with the safe capsids we have now that replacing the full copy of KCNQ2 is possible, which makes us want to think for this specific gene, the strategy of ASOs, of regulatory elements are probably going to be more promising than the idea that you're going to get an investigator or a company to just take those 72,000 base pairs, the other 1,700 to make it run and get them in a virus that's safe to introduce. There are bigger viruses. Adenoviruses, lentiviruses could probably hold those, but there's more concern that they may integrate into your genome in a dangerous way. They may provoke stronger immune responses. Again, there's a lot of smart people and a lot of companies working on this. So hopefully our limits will change in all directions, but right now this is a big limit. So this is a slide showing just one of the single guide arm. This is kind of how it works. DCAS9, remember we talk about CRISPR. This is a CRISPR enzyme that has been killed. So D stands for dead. So dead Cas9 is not a living CRISPR enzyme. It's been deactivated. It will not cut DNA itself. But what Cas9 is great for is localizing a region, and you can attach something to it like a regulatory element or promoter that turns the gene on. So you have a single guide arm here, the CRISPR DCAS9 single guide RNA. This will recognize the exact sequence of the DNA where you want it to land, because you've got 3 billion letters of DNA in each cell. This enzyme, which originally, as I'll get to, is a bacterial enzyme to kill viral invaders. And so it became the bacteria's immune system, basically. It's been co-opted by brilliant molecular biologists to do gene editing, and in this case, to do gene delivery, to deliver these little regulatory elements exactly where they want. And then if this bubbles here, it lands on a promoter and turns the gene on and can produce more genes, more messenger RNA and protein. So Encoded Therapeutics is a company that has focused on regulatory elements as a way to treat right now Dravet syndrome. I believe they're looking at others as well, but they do screening through a variety of tools to identify where these regulatory elements will be. And there are paradigms that are well known. We're looking in the non-coding DNA. These are not part of the genes. These typically precede the genes. But you can see if there's a little stretch of non-coding DNA that hasn't changed since mice to monkeys to humans, that tells you that that's doing something important. And there's been selective pressure not to change those 120 or whatever it is base pairs, but they get preserved. Another thing is the proteins that surround DNA. You can see certain patterns of those proteins or where DNA tends to get open. The DNA, the histones that cover the DNA open up and they can be translated. So again, there are a variety of clues that molecular biologists can use to identify regulatory elements. Of course, the regulatory elements for any gene, the potential candidates can be in the hundreds, if not more. So they ultimately have to do more refinement and then test if that regulatory element or that sequence that they think is a regulatory element, because it looks like one, will it drive more gene expression? And that ultimately has to be tested in a Petri dish. If it does drive gene expression in a Petri dish, then they're feeling pretty good. They may have found a real important regulatory element for that gene. So this is showing the cell type specificity. Again, for Dravet syndrome, it's mainly the inhibitory interneurons. And you can see a standard regulatory element just turns everything on all over the place. But here it's a very localized to a small neuronal population, which is what they want. And cell type specificity gives you expression where you want it, and it doesn't give you expression where you don't want it. So that's a very nice feature. And so this is a little view of what these regulatory elements do. But here's downregulation. Here's the protein itself, whether it's a bad protein that you want to shut off or a good protein that you want to turn up. But this is a way to downregulate it if it's a bad gene and upregulate it if it's a good gene. So like I said, just like lights, there are switches that turn it on and off, dimmers that turn it up and down. So the goal with Encoded, they're going to do intracerebral ventricular injection of this vector that should only go to the inhibitory GABAergic neurons, which is where we think the pathology, the problem with the SCN1A gene is in Dravet syndrome. And they've engineered this transcription factor, a regulatory element, to potently and specifically upregulate this gene. And it does it in human immortalized pluripotential stem cells that have been derived into GABAergic neurons. And it also works in the mouse and non-human primate brain. And again, the same type of thing I showed you for the Stoke ASOs. These mice, when they're given the encoded regulatory element therapy, they have less seizures, they have less SUDEP and spontaneous seizures. And this nice part of this, unlike ASOs, is it should be a one and done. In principle, once you give one injection, this regulatory element is incorporated into the DNA of those inhibitory interneurons and should be there for their life because those cells don't divide. So it is, it would be a miraculous therapy, and we hope the preclinical data, I don't think the trials have yet started in humans, but they're planned, and it'll be an exciting time. So this is some work with regulatory elements from a colleague, Navneet Matharu, and her colleague, Nadav Ahutev at the University of California, San Francisco. This is a SIM1 gene. It's expressed in the hypothalamus, a tiny region of our brain that controls hormones and the autonomic nervous system. And among the hormones and functions, it controls appetite and weight. So about 3% of morbid human obesity, not the vast majority, but of people who really get obese early in life and stay obese and nothing seems to work, about 3% of those individuals will have a haploinsufficiency of SIM1. They have one good copy and one bad copy. And the result, they can do a mouse model of the same gene. And if a mouse, this is a normal mouse with two good copies, and they look make the normal amount of messenger RNA, and they're thin, they're not overweight, they're not too skinny, they're just normal mice. If you're missing, you have one good copy and you have this mutation that makes this copy bad, these mice are obese. And if you use an activator with a DCAS9, just what I showed you previously, you can upregulate this and you get a normal mouse. And this was published 2 years ago in Science. But this is the beautiful, what you want to see. If you can actually cure a mouse, that's great. Fixing things in a Petri dish is very good, I'd say, but curing a mouse is excellent. And it's a much bigger step to feeling like you may be really onto something. And this is another group that I've been fortunate to collaborate with. They were at NYU and then they went to the Broad and Harvard. This is Jordan Demichstein's work and Gord Fischel and Bornstein Schneider. And this was published 2 years ago. This is on SCN1A. And again, similar to what Encoded did, but a slightly different approach. This is a very specific regulatory element that turns up SCN1A in parvalbumin positive inhibitory interneurons. So again, it's even more cell type specific. It's not targeting all inhibitory interneurons, but the exact subpopulation that we think is critical in Dravet syndrome. And what you can see is whether it's in the rat, in the marmoset monkey, in the macaque monkey, or in human brain tissue taken at the time of surgical resection, using this regulatory element can greatly increase expression of SCN1A. So again, we believe that this is going to become more widely adapted. I mean, no one's treated a brain disorder yet. Encoded will hopefully be the first one soon. And I hope to goodness it works because it will open the door for so many other genetic disorders. Now I'm going to turn and pivot a little bit to CRISPR, which has been a very hot area. As many of you know, there are a lot of companies, a lot of them having what are some consider crazy valuations for having collectively treated a few patients. But CRISPR are DNA sequences from bacterial genomes that were originally derived from DNA fragments of infectious bacteriophages. So literally there were little viruses, bacteriophages that would always, this battle, that goes on endlessly between parasite and host. This was the original parasite and host battle. There were viruses infecting bacteria, and bacteria used parts of the viral DNA to fight back. And then they learned to make tools to recognize the DNA and destroy it if a similar bacteriophage ever came back and infected it again. Jennifer Doudna got the Nobel Prize for figuring out that this bacterial system might be used to edit genes. It was a brilliant thought, and it's now used routinely, and I would guess thousands of labs across the world to edit human genes and either create mutations or correct mutations or skip abnormal exons, has been done as well in different gene therapies. So this is an example of my colleague Chenzhu Long, who started out working on Duchenne's muscular dystrophy, came to NYU to work on cardiomyopathies, but I was fortunate enough to meet him and convince him he should work on some neurogenetic disorders as well. And this is a child I care for with a KCNQ2 mutation. And here you can see this is the wild type. So every 3 letters, these 3 letters make glycine, these 3 letters make tyrosine, these 3 also make glycine, these 3 make aspartate. Here, this child this is the normal copy, wild type up top, and on the bottom in the 841st letter of the DNA, there's a change from G to A, which has changed the amino acid and caused the disease, unfortunately. A single letter out of billions that we have is enough to cause these disorders. So what he was able to do, make a long story short, using a very specific type of CRISPR enzyme, he was able to, number one, create the model and then fix it. So in a Petri dish, he can take nerve cells derived from this child or that have been artificially created to mimic the exact single letter mutation that the child has, and he can correct that letter with like 90% efficiency and no minimal off-target effects. So it's an extraordinarily, exciting field. One of the challenges that may come to mind is that all of your children, the vast majority, have different mutations. So you literally need a different CRISPR approach for 90% of children. Obviously, some will have the identical mutation, and you could use the exact enzyme, but keep in mind, it's not just the CRISPR enzyme. You need to do certain letter changes. These guys are magicians, way beyond my comprehension, but they can fix missing letters. They can fix two letters too many. So there's a lot of things they can fix with CRISPR, in addition to just substituting one letter for another, which is the classic way that the enzymes began. But this is an example in a Petri dish where the wild type and the mutation can be studied and the mutation can be fully corrected. So it's a great opportunity. Getting this into the brain, for me, is going to be one of the bigger challenges. And again, this is just a little bit more of a diagram of how it was done and exactly what was done. But this is an adenine base editor. This is the actual CRISPR enzyme. So unlike the regulatory elements where they used a dead Cas9, this is a live one. This is a real Cas9. It's not got the D before it. It's got an N. So this is functional. It makes a single-stranded break in the DNA. It has an editing window, and it swaps the A for a G, and it literally corrects the mutation. So again, there are many of these brilliant CRISPR people. A lot of other people just use existing enzymes that are known to do a certain thing and use the guide arm. But you need the guide arm to get exactly where you want to go, and you need the right enzyme to do the exact edit that you want done. But this has now become routine in molecular biology. And this just gives you an idea. The green peak here is the letter A, and that's the mutation, that's abnormal. And this is from neurons in the cell culture. After the adenine base editor, it's now been corrected back to G, which is what it should be. And the amino acid is corrected as well. So it's an exciting time. Again, we're not ready for prime time and treat patients, but at least we know we can do it in a Petri dish, and that is one of the first steps. So as I said, there are many multi-billion-dollar CRISPR companies, but none of them have any brain disorder in the pipeline that I'm aware of. And the reason is around delivery. We have 90 billion nerve cells in our brain. How many need to be repaired? Will the CRISPR molecule incite immune reaction in the brain? A lot of the viral vectors, if they're not directly delivered, can go to the liver. I think at least one patient died from probably getting way too much of an AAV9 delivery. Hopefully that will never happen again. This could potentially be delivered directly into the spinal fluid within the brain. Again, we'll need a lot of experiments in non-human primates to get both the efficacy and the safety down before we even consider thinking about treating a human or asking the FDA for permission to do that. So then the final topic is actually replacing genes. For some patients, a whole gene is deleted. You may have a section of a chromosome that's just gone. 1, 2, 3, 5 genes may be deleted, and there may be dysfunction in some cases on the other copy. So the only way to restore function would be to replace the entire gene, and that's happening. So there are diseases you can see on this slide. This is just a summary of some on the top where they're actually replacing these different lysosomal storage genes that are dysfunctional or absent. And then on the bottom, these are neurodegenerative disorders where they have a gene that makes a toxic protein, and here they want to shut down the gene. So again, there's gene therapies to turn things on, fully replace the gene, and other ones to shut them down. And there are current trials going on with AAV gene therapy in Parkinson's disease. Again, this slide, look, is from 3 years ago by Ben Deverman up at MIT, who's been one of the pioneers in making different capsids. And these are treatments being injected into the basal ganglia, the part of the brain that's diseased in Parkinson's. The point is, this field is evolving, and we will benefit from the lessons learned in other disorders. We're not there yet with the neurogenetic epilepsies to fully replace genes, but once there's breakthroughs in other diseases, I think breakthroughs in epilepsy will follow. And then finally, I just want to give you an idea. This is from Ben Deverman's paper, just some of I talked about these things, but what goes into that additional packaging that's not just the gene or the regulatory element. So you have to have a protein, transgene here, and then there's all these different sequences to be put in there. And this is the idea of gene replacement, where you actually replace an entire gene. But this is what the inside of that automobile looks like. The gene is the engine, but you need a bunch of other pieces to make the engine work and to get incorporated into the chromosome where you want it and expressed. This is a gene replacement for an antibody. Antibodies have heavy and light chains. So again, these are like abilities to give people antibodies they may be missing. And then these are strategies, as I said, using ways to shut down, knock down toxic or bad genes. So again, this slide is now 3.5 years old. The field continues to move at light speed. So again, just turning back now to delivery, because I think that will be our big bugaboo for brain disorders. The blood-brain barrier is one. Cell membrane, you have to get, even if you get into the brain, you've got to get condensed DNA. It's got to be packed into a small size, and it has to get into the cell by endocytosis. You don't want it coming in and then getting chewed up, so it's got to escape from degradation. It's got to move across the cytoplasm, which is slow, and it's got to get into the nucleus, which may not be so easy. There are nuclear pores, but they don't let everything in. And then the gene's got to get into the chromosome, or if there's a little episome, which is like a little bit of DNA that doesn't have to get incorporated into a chromosome, it just kind of, it's its own little functional unit. Mitochondrial DNA in our cells is a bit like this. They're kind of their own leftover from when we were bacteria, and two bacteria got together and created the eukaryotic cell, which is all plants, animals, fungi, and yeast. All of us have mitochondria that are energy powerhouses. Without those, we would look like bacteria because we'd never be able to do anything specialized and complicated. And then you've got to keep that gene in there and make sure you don't hurt other good genes. And again, just an idea, one of the things going on now in this field is something called directed evolution, where you literally try to use evolution's processes. You make your own recombinant things, and you just try out different, these are capsids, these are viral capsids of thousands of different things, and then you deliver them and see which ones grow best, and then take those and reproduce them. But they're actually using evolution, because viruses evolved, as we know from COVID-19, so quickly, but some smart molecular biologists have taken that evolutionary speed and used it to our advantage by having viruses do their own mutations to try to make us better capsids that we can get more specific delivery to the nervous system, we can get safer delivery, things like that. So, again, this field is fun to watch from the sidelines. Have my toe in the water, but it's exciting to see some brilliant people doing it. And the last part I'll comment on is another delivery system. This is from Jake Brenner at Penn and Samir Mitragotri at Harvard. They literally take your own red blood cells or red blood cells from a blood bank that's been typed in cross. They use a protein they call cell grip, and then a cargo. The cargo can be a gene, can be a capsid that's containing a gene, can be a nanoparticle. that's got a gene or small molecule, can be an antibody. And in this case, they literally hitchhike the cell grip onto the red blood cell, and then the cargo gets attached. And what you can see here is these red blood cells are literally coated with particles, the cargo attached to the red blood cell. And it's just a gorgeous idea because, this is a loose connection. So when these red blood cells, which you can see what they look like, they're discs, when they go through capillaries, which are just like what you see in pictures, they're basically tubes. Capillaries are very thin tubes, they're like my pinky, and the red blood cell, it can't go in as a disc, so it kind of gets deformed. That deformation is beautiful because that's just what biology wants. It wants to maximize the surface area to allow oxygen to go from the red blood cell and carbon dioxide to go back in so it can get it to the lungs and we can blow it off. When the red blood cell squeezes through the capillary and deforms its shape, the cargo goes off, just falls off by mechanical displacement. It's like you have something on a balloon and you squeeze the balloon through a thin tube. Whatever is attached to the balloon will come off in the tube. And that's exactly what we want. So here's an example where they are injecting something into a mouse, into the carotid artery on one side. And you can see if they just inject it in a vein, this is the amount of delivery that you get. And if you inject it here, you get literally 15, 20 times the delivery. So this is a way to potentially get, and again, here's a heat map of the right side of the brain where the delivery happened, the left side, which was only got an intravenous injection. You can get 10 and potentially in some cases up to 100-fold delivery. So I think what we'll be seeing in the future is mixing these technologies together. We're going to be taking, there's other people I know at Caltech using focused ultrasound to break down the blood-brain barrier, which again can greatly enhance the delivery of these capsids or gene therapies into the brain. You could see combining red blood cell hitchhiking with focused ultrasound and potentially get 100 or more fold delivery, which right now is probably the biggest stumbling ground. So I'm going to stop there and thank everybody for listening, and hope I didn't put too many people into outer space.
It's thrilling, actually. I'm very happy to have a wander in the planetary system because there's so much, and I think you gave a fantastic overview of a lot of different things that we can be looking at. And I find myself thinking, this is an anniversary summit for us, right? This is the 10th anniversary of the foundational research that showed that there was such a thing as KCNQ2 DEE, that it wasn't necessarily benign, that it could be not benign at all. And to see how far things have progressed in that decade is really quite astonishing. Does it surprise you to see how far we've come in 10 years?
It does and it doesn't. It's kind of like computer chips growing logarithmically. I mean, I just feel like it's so exciting, and there are just so many young, brilliant people. It's like they're speaking languages I couldn't even imagine, but I can follow the gist pretty well, and it's just exciting to watch what's happening.
Well, I only have a few questions for you because this is just, it's a super presentation. I found myself noticing a couple of times a number of, SCN1A, for example, cropped up a lot. Some other, I call them cousins in the genetic epilepsy field, right? And it's exciting to see them doing well. For those in the KCNQ2 community who are saying, oh, why not us? Is that just how long people have been in the game? Or is it really more a fact of some of these genetic epilepsies may prove to be a little bit easier to resolve than others, or both?
I think it's a little bit of both. I can tell you, I've advised companies, and some of them have these beautiful spreadsheets that they've done. And one of the things they look for is, they want genes with the right number of people. If there are too many, it won't be an orphan disease, and it's a harder FDA path to go. But you're definitely in the orphan category. They do want strong disease associations. So they want disease associations that have natural history databases. If they have that, it's going to make their job a lot easier. If there are investigators that have large numbers of these, if you can identify. So what you need to do as a community, and you may know this, and I may be preaching to the choir, you need to organize your patients. You need to have, if a company says we want to treat premature stop codons, you need to say, great, here's a list of 426 children around the world with premature stop codons. We know who their neurologists are, and they potentially, these 211 of the 426 have said they'd be interested in hearing about it. To commit to anything, hearing about a small molecule or gene therapy, or they're interested in research on a natural history study. That's what companies want, because it's going to make their path so much more likely that they can achieve success. They go to a disease where there's 4 organizations and they hate each other and they're all fighting. It's like, let's go elsewhere. It's not worth it. And if they go to one, like I'll give an example, the one that I mentioned a little bit tonight is CDKL5, where I'm very close, with the family, the Jafars from Dubai, who have a beautiful 7-year-old daughter who has this disease. And they literally, I don't know what they've done, but they have natural history studies. They have 2 gene therapy companies working on this. They've have 4 different epilepsy small molecule companies doing trials on their disease. So if you organize, and I would say they've been the most spectacular example, because Dravet's been around a while and they're kind of the old one on the block. And there's been 15 years ago, we figured out that it was the inhibitory interneuron. So there's been basic science going on. There's been clinical science there. We did the cannabidiol trial in Europe. They did steripental on Dravet. Fenfluramine just got approved for Dravet. So Dravet's got a number of small molecules, and now you see 2 gene therapy companies are in the space, and I know of several others that are working on it. CDKL5 is not as far, but they do have several, molecules, small molecules, and at least I think 3 gene therapy companies are focused on CDKL5. And part of that is because the community has really organized under the leadership of, a family and also centers of excellence. You want to have centers of excellence that really have experience with these kids and can start collaborating and doing studies for biomarkers, for natural history. That's what I would strongly recommend to the community. And then for leaders to get together and say, how do we drive this? Do we drive it through a couple academics? Do we drive it through a gene therapy company? Do we do both ideally? Because no one knows who's going to get to the finish line first.
Yeah, no, I think it's a great shout out, because we do have a natural history study, and this is a shout out everybody in our community to sign up, participate, fill in the paperwork, and we'll have more on that elsewhere in the summit. Following up on one thing that you mentioned regarding gene therapy, it's my understanding that for KCNQ2 in particular, ASOs offer more promise than gene therapy. Can you just briefly walk through? I mean, that was a spectacular presentation. Yeah. So I'm asking you to give us a bit of a shorthand explanation right now of why ASOs might be more promising for KCNQ2.
Well, I think number one, ASOs are more promising for any neurogenetic disorder because they're already working. So we already have examples where they're now working. The manufacture of ASOs is pretty trivial. Say it's nothing. You have to get the right combination of letters, and there are things that they don't break down, but the science has been worked out, and there are many, many diseases already with approved ASO therapies, and probably 50 or 70 with trials that are going on. So I think based on the size of KCN, so you're not going to replace KCNQ2 anytime soon because it's a monster large gene, and getting monster large genes into the brain, no one's done it. And I don't think it's going to happen soon. It'll happen, but it's not happening soon. So for all those reasons, I think, yes, absolutely. ASOs are almost always, if you said, a couple with a gene that's only been reported in one other person in the world, what would be the best strategy? I'd say ASOs. Start with an ASO. And there are different, as I said, there are different ASO strategies depending on whether you're deficient and you want to upregulate the gene, or whether you have a toxic protein, you want to downregulate it. But ASOs allow a lot of flexibility depending on the molecular biology of your specific gene.
One other question here, in terms of the age of people, we have a huge range in our community. We'll have people joining us who have just had a child diagnosed, and then, our daughter is 17 years old now, and we have people in our community in their 40s. So would you say that some of these treatments, be they ASOs or potentially medicines or the like, might hold promise for people of any age, or would you say that they're only going to be for younger people? What is your thought on that?
I think my thought is clearly that nobody knows. It's a $1 million question. No one knows the answer. My gut, for what that's worth, is that they will be worthwhile for everybody. And there'll be a curve. The younger you give it, if you can make a diagnosis in the utero like you can with tuberous sclerosis, that's the best time to treat. If you can get a 6-month-old fetus and treat it, you'll probably have, potentially full rescue, would be my guess. Maybe not full because the brain's already forming. But if you take someone at 1 or 2, the brain's already been developing for a period of time, but there's just a lot of opportunity to rescue. And even for a 17-year-old or 20-year-old, I mean, I tell one story of tuberous sclerosis, a child we did surgery on. That's a very different form of therapy, but he was 30. He was very autistic. He'd come into my office. He was verbal, but he was just, mommy, 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 when are we going to McDonald's? When are we going to McDonald's? And she'd say, please, please, let me speak to the doctor. Mommy, 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 when are we going to McDonald's? When are we going to McDonald's? It was like for 30 minutes, that would be the whole visit. He had surgery at age 30. He would come in the office, oh, Dr. Davinsky, how's your day? At age 30, I never thought I would see that. This person, just sprung up as like a person to interact with and speak to. Before that, it was just like an anxiety machine on replay. Not that there wasn't a person locked in there, but I couldn't really access him or see him. And after the brain surgery at age 30, it was like a miracle. So I think, all of these kids have more in them than we know. For everyone, they understand more than, they may have limited output of expression. Like, they've seen them on 60 Minutes and other people with autism I see in my office all the time who are nonverbal, and then just got a thing to point at the letters, and they speak beautifully.
Yeah,
and
we
they're
see that in our
complicated
community.
thoughts. Yeah,
Absolutely, we see that
still
so
send
much.
shivers up my spine. So there's a lot in these kids that could be unlocked with just 5% improvement in a 20-year-old could be big.
I totally agree. Getting back just quickly to the CRISPR story that you told there that specifically referenced KCNQ2, where you changed an A to a G, that's obviously extremely exciting to see that happen in the Petri dish. 2 questions on that. What's the next step for that? We'll go into a mouse model, for example, and how promising or useful is that for the broader KCNQ2 community?
So I think the next step is to do an animal model and to show if we can get it into the brain and rescue the phenotype in some way, whether it's seizure threshold or putting electrodes in and measuring excitability and getting it to be normal. And then I think these can be done individually, or there may be specific exons in KCNQ2 where a lot of the mutations cluster. This will be different for different genes, but I do think, those are possibilities as well. So I think CRISPR is a harder road to toe than ASOs for sure, but it does offer like the perfect correction. You're not adding anything, you're not deleting anything, you're just changing things back to normal. So there's a nice, harmony about CRISPR. It's just, everybody, it will take a lot of work but again, he was able to make that KCNQ2 correction in a few months. And you can imagine those things will get automated with computers and robotics. And we'll just we'll have enzymes, and we'll know based on the letters around them, what letter you want to change, what the company is, because that changes it too, what the environment is of letters on the left and the right. And there'll be 25 enzymes, and they'll just say, oh, we want to fix this one. We'll take enzyme 18, combine it here. I think it will speed up, radically with time, but that's in the future.